All right, y'all. Another week, another topic. Let's get some notes in. Roll that intro. All right, you guys, let's go ahead and look at our Unit 2 notes on Punnett Squares. All right, this week you are going to be filling out the light green box on your notes. So, Punnett Squares, guys, are just diagrams that are going to help you determine what's the probability or how likely you are that an offspring is going to inherit some certain genotype or a phenotype from two parents. Right now, you can use a Punnett Square for traits that are controlled by one gene. It's going to be two alleles, right? So let's look at this square over here on the side. You have capital T, you have little t. What could that T stand for? You know what? I think it's going to stand for tall, how tall something is, right? Because most of the time you use a letter that kind of matches up with what trait you're looking at. So maybe capital T means tall. So that means little t is going to be what? Little t is going to be short. All right. You have one gene, one trait, how tall something is, by two alleles. So tall or short. Now, the Punnett square is a square that's going to be divided up into 4 sections, and this makes it for real easy math, all right? Each section represents 1/4 or a fourth of that possible inherited combinations. Each square is 1/4, or, this is like easy math, 25%. I'm saying it's easy math because this is money math, all right? You have 4 quarters and a dollar, right? The same way you have 4 quarters in this Punnett square. So 25%, 50%, 75%, 100%, all right? Now, let's look at that bottom bullet for a second. The parents' alleles for a trait are written along the top and the left side of the square. Parent 1, parent 2, it doesn't matter which one's which. One of the parents has to go on top. One of the parents has to go on the side. All right, they've got to be on the outside of the Punnett square. And you see how I kind of took those two letters, like maybe parent 1 is capital T, capital T. I kind of spread them out to the side a little bit. And parent 2 is little t, and I kind of spread them up and down a little bit. That way there's like one letter on top or beside of each box. Now, how you're going to draw this Punnett square, all right, pretty easy. Honestly, when you take away those letters and I just look at the box, it kind of looks like a window to me. So you're going to draw a window, or it's just a square with a plus sign in it. You want 4 small squares. Now, you're going to write the genotypes for the parent. Again, parent 1 goes on top, parent 2 goes on the side. It doesn't matter if it's mom or dad, whatever. And you're going to write those genotypes. Again, spread them out just a little bit, right? Parent 1 on the top, parent 2 on the side. Now, look at this, because this is a little bit crazy, but bear with me, okay? You're going to multiply the top and the left sides of the square for each row and each column. It's kind of the same thing like when you have a multiplication table, all right? The 4 combinations inside are going to represent the 4 possible combinations for alleles for any given child or offspring. Again, now this is kind of crazy and confusing looking. Don't worry, all right? The next slide is going to help out a lot because I went ahead and made a little animation, all right? So we're going to take this. I know it's not exactly the same setup as the last one, but capital T, little t for parent 1 on top. Parent 2 on the side is little t, little t. I spread them out a little bit so that way there's one letter on top of each box and one letter to the side of each box. Now I'm going to drag those letters and I'm going to copy them to everything below or to the side. Now watch, I'm going to take that capital T, bring it down, and I'm going to copy it to the 2 boxes underneath it. It doesn't go anywhere else. It just goes whatever's on top. You just drag right down. All right, I'm going to go to the next little t on the top from parent 1. And I'm going to drag that down. You see that little t copies to the 2 boxes beneath it. All right, I'm going to take parent 2, little t. I'm going to drag that little t across and copies to those boxes. I'm going to take that last little t from parent 2, drag it across so it copies over like that. All right, that's all you got to do. I mean, you drag down, you drag across. It's like you're copying, all right, copy pasting the letters that are on top or the letters that are on the side inside the box. The main thing is that you want each box inside to have 2 letters. All right. Now, Below or to the side of the table, wherever you have room, you're going to write the possible genotypes that you could get from these parents. Now, if you look over here on the side, I've got my Punnett square, right? I don't have room on the side to write this out, so I kind of went down below and wrote it. And so I have the same thing, right? I have capital T for parent 1. Parent 2 is little t, little t. I know it's a little bit different from the previous slides. That's fine. All right. I got parent 1, I got parent 2. I drag parent 1's capital T's down, and I drag parent 2's little t's across. That leaves me with 2 letters in every box. And each box, if you look, each box is actually the same, right? It's capital T, little t. Box 2, capital T, little t. Box 3, that bottom left, capital T, little t. And the last box, capital T, little t. So they're all the same. All right, you're going to determine the probability of each genotype. Remember, each one is 25%. So how many do I got? I have 1, 2, 3, 4, right? Or I have 25, 50, 75, 100%. So 100% or 4 out of 4, all of my genotype possibilities are going to be that capital T, little t. Now I'm looking down here at the bottom where it says 0% big T, right? I don't see none of those 4 boxes inside have a big T. Same thing. I'm looking for little t. All 4 of those boxes, there's no little t. So that's why it's at 0%. There are 0 boxes with big T, or 0 boxes with little t. Now, you're going to determine the possible phenotype that could result from these parents. And remember that genotype, the letters, the genes, tell you what the phenotype, the physical appearance is. Now, if we're pretending that a capital T means tall, right, as long as there's one capital letter, that capital letter is always louder than the little letter, right? So for this example, if capital T is tall and I'm like screaming tall, or at the same time, someone whispers short. For little t, we go 1, 2, 3, and I yell tall. What are you going to hear? Are you going to hear that person whisper short? Well, maybe, but probably not. You're going to hear me scream tall. So tall is going to be that phenotype that we see. And if I flip back to that last slide, genotype, it's 100% is capital T, little t. My phenotype, same thing, 100%. What's capital T, little t? Tall. All right, that's how you're going to take this Punnett square. You're going to go from parent 1 to parent 2, fill out the square, and then you're going to have your genotype and your phenotype. So pea plant height is controlled by one gene. All right, so there are 2 alleles for this gene. Either the pea plant is going to be tall, capital T, or short, little t. Now, tall pea plants are actually the dominant. All right, so that means short pea plants are the recessive alleles. Now, the next bullet is going to be an example, guys, and we're going to kind of continue on with this example on the next slide. So 2 parents are both heterozygous, capital T, little t, and they're tall, all right, and they're crossed to produce offspring. So what are the possible genotypes and phenotypes of this offspring? Now, that kind of sounds a little confusing. Don't worry, we can actually break it down into a couple easy steps. So first off, determine what are your parents, right? 2 parents, both heterozygous, that big T, little t. All right, boom. So that means parent 1 is big T, little t. Parent 2 is big T, little t. We're going to make the Punnett square, fill it out, drag parent 1 down, drag parent 2 across. And then what are we left with is the possible genotypes on the inside. And then that means we know our phenotypes once we kind of figure out what those genotypes mean. So let's look at this next slide. 2 parents, again, both big T, little t. So I put parent 1, big T, little t on top, parent 2, big T, little t on the side. I drag parent 1 down. So I drag that capital T down. I drag that little t down. And then I'm going to take parent 2. I'm going to drag that capital T across, drag that little t across. So I have my boxes on the inside, right? So the first box is going to be big T, big T. The next box over that top right is going to be big T, little t. The next box, that bottom left, is big T, little t. And lastly, that bottom right box is little t, little t. All right, so our genotype, remember, we're going to do percentages. So how many boxes have a big T, big T? When I look back, I see 1 box has big T, and I know that means 25% big T, that homozygous tall. All right, how many are big T, little t? Well, I see 2 boxes. 2 boxes means that's 25, 50%. 50% is big T, little t, that heterozygous tall. Lastly, how many boxes are little t, little t? I see 1. That's going to be 25% is little t, homozygous short. Now, that's going to tell us our phenotype. Our phenotype is how many of them above say tall. There's the big T, which is 25%, and there's big T, which is 50%. Well, I know that equals 75%. So 75% are tall and 25% are short. All right, so let's go right back to the beginning, guys. Do a quick review. That way you can kind of copy, pause, copy, pause, copy, pause, and work your way through to get your notes done. All right, remember, here's the one. If it's a little confusing, watch the animation. All right, guys, that's it.
All right, y'all, if you learned something, go ahead and smash that like button. If you have any questions, please do not forget to message us on Schoology. And if you missed something, why don't you go ahead and click on one of those videos and get yourself caught up. All right, y'all, that's all I got for this week. See ya.